Hi guys, welcome to the discussion of Chapter 7, Mutation, Biology Semester 1, SB 015, Malaysian Matriculation System. These are the learning objectives that you need to complete at the end of the lesson. At the end of the lesson, students should be able to define mutation, state the two types of mutation which are spontaneous and induced mutation, classify mutation into gene mutation and chromosomal mutation, state and define the four types of gene mutation, state and define the four types of changes to the proteins following gene mutation, explain sickle cell anemia as an example of genetic disorder resulting from gene mutation, define mutation, and state the types of mutation and examples of each. Now let's take a look at the definitions of various terms that we will be using throughout this chapter. Mutation refers to a permanent change in the nucleotide sequence of a gene or the amount or structure of the chromosome of an organism. A mutant refers to the organism carrying mutated gene, chromosome, or having abnormal chromosome number. Mutation, however, refers to agent that causes mutation. There are two types of mutation. Firstly, spontaneous mutation. Secondly, induced mutation. Induced mutation happens when organism intentionally exposed to a mutagen. As we all know, mutagen is any agent that will cause mutation. There are three types of mutation. We have physical mutation, for example, UV ray, X-ray, and gamma rays. We have chemical mutation, for example, colchicine, ethidium bromide, and mustard gas, and also biological mutation, for example, viral DNA and transposons. Spontaneous mutation happens suddenly, for example, error during DNA replication or chromosomal non-disjunction during meiosis I or meiosis II. Chromosomal non-disjunction refers to the failure of chromosomes to separate during meiosis. For example, if everything takes place normally for this cell during meiosis, it will result in the production of gamete cell with one copy of big chromosome and one copy of small chromosome. However, let's take a look at this. There is the occurrence of chromosome non-disjunction for this big chromosome during meiosis II. This big chromosome failed to separate, causing this gamete cell to have two copies of large chromosome. Meanwhile, this gamete received no large chromosome at all. If this gamete fertilizes a normal gamete, it will result in individual with extra one chromosome, or 2n plus 1. If this gamete fertilizes with a normal gamete, it will result in the production of individual with less one chromosome, or 2n minus 1. In the case of human, normal diploid individual will have 46 chromosomes. However, 2n plus 1 individual will have 47 chromosomes. Meanwhile, 2n minus 1 will have 45 chromosomes. There are two classes of mutation. These are gene or point mutation and chromosomal mutation. Gene mutation refers to mutation that involves the changes of sequence of bases in a gene. It only involves a few nucleotide base pairs. For example, do you still remember in the assignment regarding lac operon with mutated regulatory gene? Chromosomal mutation refers to mutation that involve the changes or aberration in the structure of the chromosome or changes in the individual's overall chromosomal number. For example, Down syndrome individual having 3 copies of chromosome 21 instead of just 2 copies. This causes the overall chromosome number for a Down syndrome individual to be 47, unlike normal individuals that have a total of 46 chromosomes. Let's focus on gene or point mutation first. There are 4 types of gene mutation. These are base substitution, base inversion, base insertion, and base deletion. Base substitution refers to the replacement of one nucleotide base pair with another nucleotide base pair. For example, this is the original DNA strand, and this is the DNA strand after mutation has happened. As you can see, everywhere else is the same except for this base. After mutation, this base has been replaced by another base. In this case, the original adenine is being replaced by guanine. The next type of gene mutation is base inversion. Base inversion refers to 2 nucleotide pairs which are side by side becomes inverted in the DNA sequence of a gene. For example, this is the original DNA sequence for the gene. And this is the DNA sequence after mutation has happened. Everywhere else is the same before and after, except for this G and A, which are located side by side. After mutation, this G and A has become inverted in sequence and become A, G. The next type of gene mutation is base insertion. Base insertion refers to the addition of an extra pair of DNA nucleotide to the existing DNA sequence in a gene. For example, this is the original DNA sequence of the gene, and this is the DNA sequence after mutation has taken place. As you can see, an additional adenine has been added in between A and G. Once this happens, it causes the base G, C, 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 T, and all the subsequent DNA nucleotides to be pushed back. The last type of gene mutation is known as base deletion. Base deletion refers to the removal of a pair of DNA nucleotide from the existing DNA sequence in a gene. For example, this is the original DNA sequence of a gene. This, however, the DNA sequence of the gene after mutation has taken place. As you can see, one of the cytosine in the original DNA sequence has been removed. Once this happens, the base T, A, T, G, and all the subsequent DNA nucleotide will be brought forward. The various gene mutations could potentially have an impact on the protein that the gene codes. There are 4 effects of gene mutation on proteins. These are silent mutation, missense mutation, nonsense mutation, and also frameshift mutation. Since one amino acid can be coded by more than one codon, as you can see in the codon table here, sometimes changes in the nucleotide base sequence in the gene will not result in the change of sequence of amino acid in the polypeptide chain. For example, this is the original DNA sequence of a coding strand of a gene. This, however, is the DNA sequence after mutation has taken place. As you can see, the base adenine in the original DNA sequence is being replaced by the base guanine. However, since GAA and GAG both code for glutamic acid, the protein produced after gene mutation remains essentially the same. This is known as silent mutation. Sometimes, changes in nucleotide base sequence in the gene will result in the change of sequence of amino acid in the polypeptide chain. For example, this is the original DNA sequence for the coding strand of a gene. This, however, is the DNA sequence of the gene after mutation has taken place. As you can see, the original adenine has been replaced by cytosine. The codon GAC codes for a completely different amino acid compared to the codon GAA. In this case, coding for aspartic acid instead of glutamic acid, so therefore producing a slightly different protein. This type of change in the protein is known as missense mutation. Sometimes, change in the nucleotide base sequence in the gene can result in the appearance of a stop codon in the middle of the gene. This causes the resulting protein produced to be truncated. For example, this is the original base sequence of the coding strand of a gene. The substitution of base guanine here to thymine causes this codon to change into a stop codon. Due to this, translation process will stop earlier than where it should be, producing a shortened protein. This type of change to the protein is known as nonsense mutation. Base addition and deletion will cause the shift in the codon reading frame, resulting in the production of a completely different protein from the point of mutation. For example, the removal of guanine here causes all the subsequent bases to be brought one base backward. As you can see, G here is located in the next codon is now shifted into this codon. C, which is located in this codon, is now shifted into this codon. So therefore, this will result in the production of completely different sequence of amino acid behind the point of mutation. This type of change on the protein is known as frameshift mutation. Now let's do a little activity. Given the template strand of the gene for hormone X have the following base sequence. After transcription, this is the sequence of the mRNA that we will get, and after translation, this is the sequence of protein that we will get. Now let's say if gene mutation occurs causing the gene sequence to become as follows. Now you have to decide what type of gene mutation is this. What type of mutation occurring on the protein? Decide if the codon reading frame on the mRNA has been changed or not. And decide if any of the following has occurred on the protein. No change, change just one amino acid, change the whole sequence of amino acid from the point of mutation, or produces truncated protein. The first step is for you to compare the original DNA sequence with the mutated DNA sequence. As you can see, the base guanine has been added in between the two cytosines. Due to this, all the subsequent bases are pushed one base forward. This type of gene mutation is known as base insertion, and the effect on the protein is known as frameshift mutation. Step 2. Transcribe and translate the mutated DNA. Transcription of the mutated DNA sequence will give you the following mRNA sequence. Translation of the mutated mRNA will give you the following amino acid sequence. Now let's compare between normal protein with the mutated protein. As you can see, all the amino acids beyond the point of mutation has changed. Now let's answer. This type of gene mutation is known as base insertion. The effect on the protein is known as frameshift mutation. Does the codon reading frame on the mRNA change? Yes, it does change. As you can see, as an additional guanine is added in between the two cytosines here, this causes the subsequent nucleotide bases to be pushed one base forward, thus shifting the codon reading frame. For example, the adenine here is now pushed to the next codon. The adenine here is now pushed to the next codon. The adenine here is pushed to the next codon. The thymine here is now pushed to the next codon, and so on and so forth. Now highlight what happened to the resulting protein. As we can see, this type of gene mutation changed the whole sequence of amino acid from the point of mutation. Now let's try another example. This is the sequence of mutated DNA. First, let's compare the mutated DNA sequence to the original DNA sequence. As you can see, the base guanine here has been replaced by the base thymine. So this is clearly base substitution. Let's transcribe this mutated DNA. Transcription of this mutated DNA will give rise to the mRNA with the following sequence. Translation of this mRNA will give rise to a polypeptide chain with the following amino acid sequence. Now let's compare between this protein with the original protein. As you can see, the amino acid sequence remains the same even after the base sequence in the gene has changed. So therefore, we can say the effect on the protein following this gene mutation is silent, or silent mutation. Now let's answer the question. This type of gene mutation is known as base substitution. Since there is no change in the protein, so therefore the effect on the protein is known as silent mutation. Does the codon reading frame on the mRNA change? No, it does not change. Base substitution or base inversion will not result in the change of the codon reading frame. Now let's highlight what happened to the resulting protein. As we can see, there is no change to the protein. I hope this guidance will help you to do the activity in your worksheet. Now let's take a look at an example of disease caused by gene mutation. This disease is known as sickle cell anemia. As you can see, sickle cell anemia is caused by base substitution, whereby the base thymine in the original template strand of the hemoglobin gene is replaced by the base adenine. This causes the codon on the mRNA to change from GAG to GUG, subsequently causing the amino acid to change from glutamic acid to valine, producing an abnormal hemoglobin. As you can see in the diagram, normal hemoglobin forms globules, meanwhile abnormal hemoglobin forms fibers. This fiber will alter the shape of the red blood cell, causing it to be less efficient. Sickle cell anemia is caused by base substitution, whereby the base thymine in the normal DNA template for hemoglobin gene is replaced by the base adenine. This causes the codon on the mRNA to change from GAG to GUG, results in the amino acid to change from glutamic acid to valine, thus forming a slightly different hemoglobin. Since a slightly different protein is produced following gene mutation, this type of mutation of the protein is known as missense mutation. The resulting hemoglobin clump together forming a fiber and become stiff in nature. The red blood cell will become sickle shape and become less flexible to go through narrow blood vessels and tend to form clots. The symptoms of sickle cell anemia includes paleness, fatigues, and shortness of breath.